So now I would like to walk through with you to show a few examples of the research results. If you can follow me over here, right here, we are going to just 4 different shoes in this control, and then we just inoculate it with the Xylella. This is a little bit late, but you can see wherever we inoculate the Xylella shows symptom. This is another positive, and this is another positive. Earlier I was telling you about the leaf scorching right here, and then these are the leaf scorching. I gotta tell you, as a plant pathologist, when we see positive showing that kind of symptom, that makes us exciting. It's because we wanna see first our positive control is working, so we can compare rest of the treatments. So these are the 4 shoots that's showing. Early in the season, these pictures or symptoms show so beautifully because we were late in this year, but next year we are going to have this meeting a little bit earlier than this year, so we are going to see better symptoms. But still, you can see the symptoms that I was telling you about it. Leaf scorching, matchsticks, that you can see the leaf just left there, and you can even see islands on some of those shoots that you can see. And then fruit shriveling. It's very characteristic symptom in this one disease positive. No, nothing controlled the other. Disease positive. You can see all symptoms in this. Okay, let's go compare with the next one. So this is treatments, is the healthy control. So in this treatments, we didn't apply anything. So you can compare between the symptoms. There is no symptoms on this, and then that one. You can see some reddish color. Those are the viral symptoms. So you gotta get familiar between the Xylella or Pierce's disease versus viral diseases. Now there are a lot of virus diseases in California. They show these kind of symptoms. Anyway, this is our the negative disease control. There is no pathogen whatsoever. This is the biocontrol one that shows the good control of the disease. So what we did on those trials is that Molly, would you like to tell us how you inoculated these?
Sure. So to begin the trial, we get all the vines their respective treatments, and like Annika and Akif said, we did that with the pulse Xyloject between the second and third, or the first and second node, and you might be able to still see that marking. A week later, we came back and did the drop puncture with the Xylella, and then a week after that, we did the second dose for most of the treatments. And then for the rest of the summer, the Xylify phage product, we did that throughout the summer, once a month, until August when we started to rate. So if you wanted to see the markings at all, just everything happened between the first and third node.
When you get close, you can see the dose marks. Again, these are the main, the application point. These flags are the one that we chose applied to those biocontrol agents. Let's go to the next one in here. Pink black stripe. This is the combination of the biocontrol agent 1 and phage. Again, so this is the one we chose 4 shoots that you can see with the green label, and I don't see any symptoms of the Pierce's disease whatsoever. When you look at the results, a little bit in the back, that each one of those treatments itself seems to be working and then doing some kind of the disease control. But the combined effect, as other researchers were mentioning about it, the biocontrol agents and phage together gives you the better disease control comparing with the disease health control. For example, when you look at this graph right here, the one on the left, red one, gives you the percentage of the foliar symptoms. It's very high. Those are the disease positive. Very on the last one, healthy control, there is no disease. So between that, closer to shorter the bar is the better, that you can compare which treatment work better comparing with the control that I showed you the very first. Okay. In this blue, phage with the label rate that we applied. Okay. So there are a couple of the leaves that shows symptoms here, but again, when you look at with the results, it gives significant amount of the disease control comparing with the positive control. Just I will focus on the 4 shoots that was labeled that we also inoculated. This right here is the phage itself with the comparing with the controls, like the applied with the other controls, not the label rate. So that means we applied this one in May 3rd. Then after that we inoculated with the pathogen again. So this is the another one. I just pointed out to you a few of them. So the way that we designed this experiment is that we repeated each treatment 10 times as a randomized block design. And you will find this map on the handouts. You are free to walk around, check any of the treatments yourself, and that's all we wanted to show you. Again, this is very late showing, and then as a short notice to show you this trial. And when we got these preliminary results, again we got excited, and Molly did a good job, and then because of that she got the heavy lifting on this project. We just wanted to have a quick field day to share these preliminary results with you. So next year we are going to have same field day a little bit early in the season that you can see those symptoms better. Any questions?
So it looks like for most of the treatments you pre-inoculated with the biocontrol and the phage, except for one treatment where you let the Xylella get a head start and then tried to treat with the phage. Is that based on observations in the greenhouse that you need to pre-inoculate with the biocontrol agents to get good control? As you have heard from the Steve and Philip, so these biocontrol agents mostly work as increasing the defense mechanism of the plant. Before we get into that, we just wanted to put them into the plant to get plant ready for the pathogens. So once the pathogen that's introduced, that's the way of mode of action of those products. However, phage requires to find the Xylella to control. So that's why we think that, hypothetically think that both those biocontrol agents, phage, work together. One of them is increasing the defense mechanism, other one is targeting the bacteria disease itself. That's the hypothetical way of course, we don't know what's going on exactly. We are trying to understand the mechanism of it. That's the idea behind of that experiment. Anything else? Okay, so we have time. Feel free. We have drinks and sunscreen there, and then go check out the rest of the experiment and let us know if you have any questions.
Quick question.
Of course.
Are you testing them for the other viruses? So red blotch, leaf roll.
Actually,
does that interfere?
No, they don't interfere. Actually, we know what viruses are in this block. We know each one of them. When you are familiar with the symptoms, you will distinguish between the Xylella Pierce's disease versus Pierce's. For example, just behind you, look at this is the one that was positive control actually. Perfect symptoms. Textbook symptoms, exactly. That's why when we see those results, we said, we gotta share this information with others, and then we can show. This is like perfectly showing the shriveling. I don't see the island, but yeah, I see a little bit over here, and then matchstick, and then discoloration. This is another positive control. Okay, so we have maps, and feel free to check it out, and then let us know if you have any questions.